Hello, and welcome to this short overview on the manufacture of in vitro transcribed messenger RNA for therapies and vaccines. I'm Nathan Spangler, Senior Director of Innovation and Strategy for Aldevron's messenger RNA services platform. And in my talk today, I'm going to cover manufacturing considerations for preclinical to clinical stages of development. My perspective on this comes from contract manufacturing for advanced therapies and vaccines, which Aldevron is well known for, starting more than 2 decades ago with plasmid DNA production. That was soon followed by recombinant proteins and most recently messenger RNA. In our 20 plus years, we've accumulated experience manufacturing a wide range of products from critical starting and raw materials to drug substances. Which brings me to the topic of this talk, manufacturing messenger RNA for therapies and vaccines. Although messenger RNA has been studied for decades, its promise to be a transformative technology changing the future of medicine wasn't realized until the recent success of the COVID vaccines. The rapid transition from promise to reality created unprecedented demand for manufacturing in an industry unprepared. It's also intensified efforts to advance mRNA pipelines, further adding to pressures. Therefore, many of the challenges we deal with stem from the newness of mRNA in manufacturing for clinical use. Compared to better known technologies, such as monoclonal antibodies, there's little GMP manufacturing experience and fewer mRNA-specific tools and technologies for scale-up, which makes it hard for manufacturers to stay ahead of rapidly evolving needs. But before I get into manufacturing, let's first talk about messenger RNA for therapies and vaccines and how properties of this molecule might impact manufacturing. The two most common forms used are conventional non-replicating messenger RNA and self-amplifying messenger RNA that incorporates an engineered alpha viral replicon. The viral replicase genes will direct intracellular message amplification, and the region encoding the antigenic or therapeutic protein will be expressed as a separate entity at high levels. This self-replicating activity means that a desired level of antigenic or therapeutic protein expression can be achieved at a lower dose and a potentially more favorable safety profile. This also means smaller scale production and less demand for expensive and scarce materials. With both forms of messenger RNA, which I'll generally refer to as mRNA for short, the 5' cap structure, 3' poly A tail, and untranslated regions are important for maximizing expression and stability. Manufacturing either type of mRNA by in vitro transcription involves the same methods and materials, but process details will vary, partly because the self-amplifying constructs are very large and require special care to maximize product quality and process yields, which I'll talk more about shortly. The applications for mRNA are many and diverse and include protein replacement therapy, gene editing, stem cell reprogramming, immunotherapy, and infectious disease vaccines. And each application will have distinct requirements for product attributes, impurities, and contaminants. For example, a protein replacement therapy will call for sustained expression without activation of an innate immune response, while a vaccine might require the opposite. A high-dose mRNA therapeutic might have much lower limits for contaminants and impurities than a lower dose application, such as a vaccine, based on self-amplifying messenger RNA. Such a wide range of requirements, in addition to the variable physical properties of mRNA, calls for manufacturing solutions tailored to fit. The manufacture of mRNA by in vitro transcription begins with a high-quality DNA template, and the design of that template incorporates elements needed for mRNA manufacturing and for mRNA function. The template construction approach that we like to take for conventional messenger RNA is to synthesize the entire template from T7 promoter up to the poly A tail and insert this into a high copy backbone that adds the poly A feature to the 3' end of the template. Such an approach avoids introducing a cloning scar in the 5' UTR, and it avoids unpredictable delays due to the difficulty of synthesizing a poly A trap. For those starting with only a coding sequence, we offer conventional and self-amplifying messenger RNA backbones that provide the flanking elements for a complete template design. Note that self-amplifying constructs will be larger and more complex, with additional elements for message amplification and expression, as shown here for one of the pre-configured backbones we offer. The next few slides will go further into template design, focusing on conventional messenger RNA technology. When designing a DNA template to guide mRNA synthesis by in vitro transcription, there are 6 key elements to consider, and these generally fall into 2 categories: elements needed for manufacturing and elements needed for function. Let's look at manufacturing first. To make messenger RNA by in vitro transcription using common manufacturing methods, the plasmid DNA template must incorporate a bacteriophage promoter and a linearization site for runoff transcription. T7 RNA polymerase is commonly used in manufacturing to drive in vitro transcription, and the T7 promoter sequence in the template is often followed by triple G, the first 3 bases transcribed. But keep in mind that the transcription initiation sequence must be compatible with the 5' capping technology. If co-transcriptionally capping with a trinucleotide GAG cap analog, for example, the DNA template must have an AG transcription initiation sequence after the T7 promoter. To linearize the plasmid DNA template for runoff transcription, there are several important considerations. Ideally, the linearization enzyme will be widely available, economical, and manufactured animal origin free. The enzyme should leave a blunt end or 5' overhang to avoid unwanted transcriptional products that can result from a 3' overhang. The linearization enzyme should cut the plasmid template only once, but a secondary site on the backbone can be okay if it's not close to or within the template region. And finally, if the DNA template encodes the 3' poly A tail, the restriction enzyme should cut cleanly after poly A without leaving behind backbone residues. BSPQ1, for example, is a popular choice that can satisfy all requirements. Next, let's look at the template elements that can impact the efficacy of mRNA therapies and vaccines. In cells, mRNA is translated and degraded, and the untranslated regions can incorporate regulatory elements affecting the rates of translation and degradation. The 5' UTR should minimally have structural elements recognized by mRNA binding proteins to ensure proper initiation. An effective design can be minimal with only a Kozak sequence for translation from the correct start codon, which we commonly see, and other elements can be incorporated to enhance expression. A 3' UTR design can incorporate elements affecting transcript stability, expression, and localization, or a template design might only have a poly A tail following the coding sequence. Having the poly A tail template encoded makes it easier to achieve consistent and precise tail lengths by in vitro transcription. It's generally understood that longer tails increase transcript stability, but template encoded poly A tails longer than 120 bases become impractical to manufacture due to the increased likelihood of shortening by recombination when the plasmid template is propagated in E. coli. For very long tails, post-transcriptional polyadenylation using a poly A polymerase can be a good manufacturing option, but this method results in an average instead of an exact tail length, and tight process control is needed for batch-to-batch consistency. A number of process parameters will be important here, such as reaction time, temperature, and concentrations, as well as the quality of the poly A polymerase. For expression of the antigenic or therapeutic protein, design and optimization of the template coding sequence depends on what's being expressed in the use requirements. For example, codon optimization might make sense for heterologous gene expression. Uridine depletion could reduce stimulation of an undesirable innate immune response. Expressing more than one protein from a single transcript could involve the incorporation of functional elements, such as a virus-derived 2A peptide sequence. And for self-amplifying mRNA, there will be many additional elements, including the viral replicon genes that must be expressed to amplify the transcript, as well as a subgenomic promoter to express the antigenic or therapeutic protein payload. An mRNA manufacturing campaign might also involve master cell bank generation and production of the DNA template starting material, which Aldevron offers, as well as the enzymes for in vitro transcription, post-transcriptional capping, and post-transcriptional tailing. According to sponsor requirements, these critical materials can be made by Aldevron in full GMP or more economical quality grades that are appropriate for ancillary and critical starting materials that will be used for drug substance manufacturing and early stage clinical development. For therapies and vaccines in development now, mRNA manufacturing covers a wide range of scales. Preclinical batch sizes range from milligram to tens of grams, and clinical batches can fall anywhere between 100 milligrams and 100 grams. Adding to that the diversity of application requirements and variable physical properties of mRNA means that a one size fits all production process does not exist. Manufacturing must accommodate a range of methods and technologies in the upstream and downstream processes. For new manufacturing programs, process designs tailored to fit begin with questions to assess what might work best to begin acquiring process knowledge. What's the intended use? Cell therapy, protein replacement, vaccine? Is activation of an innate immune response a concern? What are the requirements for mRNA purity and activity, and the tolerances for impurities and contaminants? Double-stranded RNA impurity is a good example. In uses where tolerance is very low, the downstream process may require a polishing step to remove it. The construct design also guides and limits manufacturing options. For example, is the poly-A tail template encoded? Is the transcript designed for a trinucleotide cap analog? What's the transcript length? Answer to these questions and more will help determine the best production path, as well as materials and methods. In scaling up, some degree of upstream process optimization might be needed to improve performance and results, especially for long constructs. The in vitro transcription and capping reactions for mRNA synthesis are complex, and many factors can come into play in trying to maximize process yield and quality. We like to start simple by first evaluating a limited set of conditions that we found work well for moderately sized or very long constructs. Reaction temperature is a key variable guided by construct size, as shown here for the IVT reaction using a T7 RNA polymerase to synthesize a 7 kb transcript. In this case, the mRNA integrity was better at lower temperatures, and we've found this to be generally the case with constructs longer than 5 kb. With shorter transcripts, however, we've observed that higher reaction temperature can significantly increase reaction yield without compromising quality. In this example, for the post-transcriptional enzymatic capping reaction, we saw a positive correlation between mRNA activity and the enzyme concentration and reaction temperature as assessed by target protein expression on the surface of cells using flow cytometry. This result suggested that higher enzyme concentration and reaction temperature gave the highest capping efficiency. The optimal reaction condition compared favorably to co-transcriptional addition of Cap1 using an analog with better than 90% efficiency, according to the LCMS analysis by the provider. But as mentioned, results can be construct specific, especially when large size is involved, as shown next. Here, we found a negative correlation between the capping reaction temperature and self-amplifying mRNA activity. In this experiment, reported protein expression was measured in transfected 293 cells. Better results were obtained from self-amplifying mRNA capped at a lower reaction temperature, which in data not shown appeared to maintain transcript integrity better than the higher temperature reactions did. This illustrates a challenge faced when working with very long transcripts, which are generally more susceptible to degradation. mRNA yield and quality can also be affected by the construct or by the concentration and quality of the reaction components. And in difficult cases, a more extensive investigation might be called for to understand a construct-specific relationship between multiple input variables in the upstream process. The concentrations of the many buffer components, raw materials, and process enzymes can have significant impact on reaction yield and RNA quality. And we've seen how materials from different sources don't always perform the same. The downstream process design and development for mRNA manufacturing will be guided by its intended use, release specifications, and impurity profiles, which will vary from one construct to the next. Product and process-related impurities, such as double-stranded RNA, truncations, fragments, and DNA template, have properties similar to mRNA, which makes removal of these difficult. Manufacturing scales also vary widely, from personalized medicine to infectious disease vaccine. mRNA constructs for therapies and vaccines are very large and, as mentioned, sensitive to shear stress and degradation. This is especially true for self-amplifying mRNA. And because of its large size, mRNA does not interact well with conventional chromatography resins that are widely used in biologics manufacturing. Given the challenges outlined on this and the previous slide, combined with the lack of experience manufacturing mRNA in general, the purification solutions for use in large-scale GMP are somewhat limited, which has caused reliance on suboptimal lab-scale methods. Fortunately, manufacturing solutions are emerging as the industry gains process experience. Current technologies, such as monolithic columns, provide scalable solutions for shear stress and slow absorption to better maintain mRNA integrity. And newer alternatives, such as fiber-based resins in development, promise high capacities and flow rates with reduced process times and costs. Difficult contaminants can be dealt with using an oligo-DT affinity column, which enriches the purity of polyadenylated transcripts in the final product. This is a solid option when high purity and quality are called for at any scale. And for uses demanding the highest level of purity, polishing steps such as hydrophobic interaction or reverse phase chromatography will further reduce levels of processed proteins, endotoxin, and double-stranded RNA. The use of reverse phase, however, involves flammable solvent, which creates inhalation and fire hazards. To safely contain these hazards, a specialized manufacturing facility is required, which Aldevron built specifically for this purpose. Achieving the highest level of purity is costly, and this might not always be justified. In cases where impurities are relatively low in the upstream process and where the end use doesn't require the absolute highest purity, a simple downstream process, such as flow-through ion exchange followed by tangential flow filtration, has worked fine. The advantages to this include higher process recoveries and lower process times, which help reduce costs and maintain mRNA integrity. In the few minutes remaining, I'd like to touch on analytical methods. Messenger and self-amplifying mRNA represent a relatively new alternative to traditional platforms for vaccines and advanced therapies. And because of this, there's been less clarity regarding the test and specifications needed to ensure regulatory compliance and acceptance. Safety and efficacy are essential to the success of any vaccine or therapy. Platform and product-specific analytical methods that can measure critical quality attributes must be established as an integral component of CMC. We need tests that are quantitative, robust, and rapid to ensure smooth progression through clinical phases leading up to regulatory approvals and commercialization, and for smooth technology transfer between partners. The diverse and broad range of applications for mRNA-based therapies and vaccines adds to the difficulty of establishing standardized release tests. With so many uses, there can be a wide range of release specifications for product-related impurities, process residuals, and critical contaminants. Endotoxin is a good example where we've seen the limit as low as 1 EU per mg of mRNA drug substance in some uses, while others can be more than an order of magnitude higher. Lack of clinical experience with mRNA also poses a challenge for setting release specifications on product-related impurities and product attributes. For example, how much double-stranded RNA is a problem? Below what level of capping efficiency is the product unacceptable? A few years ago, release tests for RNA drug substance in early stage clinical development were relatively basic. A QC release panel might have only had tests for RNA content and identity, process residuals, and critical contaminants. Since then, the regulatory guidelines and industry standards for quality and testing have evolved rapidly, and we now have draft guidelines that were recently published by the USP to standardize quality tests and the test methods. Consistent with the trend we've seen in the industry, the guidelines call for more comprehensive testing of product attributes and product-related impurities that are essential to efficacy and safety. For example, the 5' cap and 3' poly A tail, which are important to function, can be measured by HPLC or LCMS methods. These methods can also quantify the level of RNA with unreacted 5' triphosphate, which is a safety concern in some uses. HPLC is quite versatile as it can also serve as a robust method for mRNA integrity and identity by retention time. Double-stranded RNA impurity, another safety concern, can be readily detected using antibody-based methods, and there are efforts underway to make this test more robust and quantitative. mRNA potency is obviously important, but doesn't lend itself well to a platform test. Typically, each developer has their own assay using a self-retranslation system or cell-based method to measure mRNA translation activity. Tests such as these for critical quality attributes are product specific, and the cost and time to implement these for release testing must be carefully evaluated. Each new mRNA manufacturing program has unique requirements. Manufacturing and release tests can be accomplished using a CMO's platform processes or product-specific processes developed jointly with the partner, or processes transferred from the developer to the CMO. For new programs, there are typically 4 steps on the path to clinical grade mRNA. Work begins with process design and exploration, or tech transfer, and the amount of time and effort here depend on program readiness for scale-up. By step 2, several things are happening in parallel. Work is underway on analytical methods as master cell bank and scale-up process development begin. If things go well, analytical methods will be fit for use or qualified, and process development completed by the time the DNA template has been manufactured. So the mRNA engineering and GMP manufacturing can follow without delay. So how much time does all this take? In general, about 8 to 9 months is shown here. And this timeline has been reduced by as much as half for urgent programs. Because mRNA manufacturing by in vitro transcription uses purified recombinant enzymes instead of cell culture, manufacturing is simpler, more scalable, and more easily expedited than for other types of biological medicines. Aggressively short timelines are becoming more common, and this can be a challenge because the duration of early steps on the critical path can vary depending on program readiness, scope, requirements, and unforeseen difficulties related to the mRNA construct designs and properties. Supply shortages add another timeline risk due to the unprecedented demand for materials as well as unforeseen disruptions. So supply chain risk mitigation has become an important element of planning and execution. In this regard, the ability of the mRNA manufacturer to also produce critical materials such as the DNA template and process enzymes under the same roof is a big advantage. mRNA-based therapies and vaccines hold considerable promise, which is being realized as pipelines enter the clinic and by the rapid development of mRNA vaccines against COVID-19. At Aldevron, we're very excited by its potential use in advanced therapies, and we're preparing for the future by building flexible manufacturing platforms to support preclinical through commercial stages of development. By early next year, we will have a second specialized suite for large-scale manufacturing with flammable solvents and for manufacturing products such as oncolytic vaccines requiring a BSL-2 environment. As a manufacturer, Aldevron stands apart by making under one roof the DNA template and process enzymes needed for self-reproduction of mRNA. For more than 2 decades, our mission has been to help improve lives through scientific excellence, and our process development and manufacturing capabilities have supported the innovative therapies and vaccines that will transform medicine. The biopharmaceutical industry is growing exponentially, and we are ready and excited to help. Thanks for joining me today. If you have questions, I'd love to hear from you.